Welcome to Saitechinfo channel, my dear students. In this session, we will be discussing about biomolecules. When we say biomolecules, there are 5 important compounds. They are carbohydrates, proteins, then enzymes, vitamins, nucleic acids. So these are the fundamental biomolecules. Carbohydrates, that is nothing but sugar. They essentially consist of CHO unit, that is carbon, hydrogen, oxygen. They can be polyhydroxyaldehyde or ketone, and based on that we can have the aldose and ketose. Aldose, os means it is the suffix for sugar. Okay, note down. Os means sugar. When I say aldose, means that sugar consists of aldehyde. So that is called aldose. Do you understand this? Os is sugar, ald represents aldehyde. And what about the other groups? Generally they are polyhydroxy. Hydroxy means OH group. In alcohol we have this hydroxy group, as well in enol. That makes the difference. Actually, enol is different from alcohol. Alcohol is the hydroxyl group attached to saturated carbon atom, whereas enol is, so this is alcohol. Enol is the hydroxyl group attached to unsaturated carbon atom, that is known as enol. Now we are going to talk about the hydroxy unit. It means it may have 3-hydroxy, 4-hydroxy, etc. Actually, it starts from polyhydric alcohol. For instance, we would have already studied about these polyhydroxy compounds. Like we know this is monohydroxy methanol, is it not? Methanol, ethanol, propanol, etc. And if you have a compound like CH2OH, then it is dihydroxy, which is nothing but glycol. Similarly, we can have trihydroxy derivatives or groups. So CH2OH, CHOH, CH2OH. Can anyone name this compound? Anyone? Unmute and tell me. That is glycerol or glycerin.
Yes, sir.
Okay. So likewise, we have even 6 carbon atoms can come together to form a polyhydric alcohol. Likewise, we have so many types of alcohol, and sorbitol is one of the important polyhydric alcohols. I'll be discussing about this later. So one thing is clear, that is, it can form a straight chain like this, and you may have OH group on the sides like this. So based on the hydroxy group, we can classify polyhydroxy or monohydroxy. So this is perfectly polyhydroxy. Even glucose itself is a polyhydroxy group. It can be a polyhydroxy, but terminally it may possess some CHO, and that is why it's called aldose. And since it consists of 6 carbon atoms, so it is called aldohexose. So what do you mean by ose? This is sugar. Hexa means 6 carbon atom. Aldo, so that is one potential aldehyde group. Okay, so likewise we name. Similarly, we have fructose, that is a ketohexose, and it may combine together to form Oligosaccharide, disaccharide. So monosaccharide, generally glucose and fructose are called monosaccharide. Saccharide means sugar. It may combine together to form disaccharide and trisaccharide and finally polysaccharide also. So these ditrisaccharides are called oligosaccharide. Okay, right, let me come back. So they are classified, as I already suggested, as monosaccharide, disaccharide, trisaccharide, polysaccharide. These disaccharide and trisaccharide together, they can be called oligosaccharide also. So monosaccharide, they have the single unit of aldehyde, for instance glucose and fructose, whereas disaccharide double units of CHO. So sucrose, maltose, when I say CHO, not aldehyde, this is the carbohydrate. So monocarbohydrate and dicarbohydrate, that is disaccharide. Don't think that this is aldehyde. CHO means that is carbohydrate, that's all, carbon, hydrogen, oxygen. It consists of pairs or two sets of carbohydrate. So that is sucrose, maltose. For instance, glucose and fructose together can form sucrose. Sucrose is nothing but cane sugar. Similarly, maltose, cellobiose. So these are all disaccharides. Oligosaccharides, as I already repeated, that is, they have di and tri units. Then poly, they have many fundamental units of carbohydrate, like in the case of starch, cellulose, etc. Okay. So if you have got any doubt, you can ask me. Otherwise, we will move to the next one. Shall I move? Gokul. Any doubt?
No, sir.
Right. Next. Now, the suffix for sugar, is dash. What is that? Anyone? Just now I told you. What is the suffix for sugar? Which is nothing but ose, is it not? Take down. So very important. So ose means sugar. Any enzyme that attacks ose can be called ACE. For example, when an enzyme, what is enzyme? Biocatalyst. When an enzyme attacks ose, it can be called ACE. Aldose, when it is attacking aldose, then it is named as aldase also. So ose, the respective enzyme that attacks ose, can be called ACE. Okay, right. Then aldose, the sugar having aldehyde group. What about ketose? Yes, it should have the potential keto group. So here we have the potential aldehyde group and here keto group. Then carbon atom having 6 units, that is 6 carbon atoms. A sugar having 6 carbon atoms can be called hexose. Okay, right. Similarly, see, that is we have got one glucose, another glucose, and there is a bond. This bond is known as glycosidic linkage. Okay, so the linkage between two sugar units, that is monosaccharide units, can be called glycosidic linkage. Okay, please note down this glycosidic linkage. And usually we can break this bond, and that is why we used to say the breakage of glycosidic linkage. Okay, right. And thereby we can get back the monomers. If it is a dimer, we can break the glycosidic linkage so that we can get the monomer. Then proteins. So far I have just given a briefing about carbohydrate. Now let us move to proteins. Proteins are the polymer of about 20 different alpha amino acids. Let me explain about this alpha amino acids. Alpha amino acid, we know acetic acid. What is the formula? Shreya, what is the formula for acetic acid? Pavani. So CH3COOH, that is acetic acid. And IUPAC, ethanoic acid. Take down. IUPAC is ethanoic acid. Now this COOH group is called the functional group, and the carbon atom attached to the functional group is called alpha carbon atom. Do you understand? For example, I have given here only one carbon atom. It may contain many carbon atoms also, like C, then COOH also. So in that case, the first one is alpha, second one is beta, third one is gamma, then delta, omega, etc. So especially the hydrogen present in the alpha carbon atom, that is substituted by amino group. For example, let me consider this acetic acid. So this is alpha. So I can write down this as H, then COOH. So here I can replace this hydrogen by an amino group, and this is known as alpha amino acetic acid. Do you understand this? Whereas this amino group can also join in the acidic proton. It can replace the acidic proton, but there it takes another name. For example, let me take the same CH3COOH, but instead of this OH, I can introduce NH2 by the reaction with ammonia, so that I am able to get CH3CONH2. So what do you call this compound? Anyone? This is not amino acid. This is called acetamide. Take down. This is known as acetamide. So IUPAC is 1,2-ethanamide. Do you understand? So amide is different from amino acid. Here it has lost its acidic property because of the replacement of OH, whereas here the acidic functionality is still maintained, and that's why still is acid. Of course, it is substituted. Where it is substituted? Alpha substituted. So it's called alpha amino acetic acids. Do you understand? And this is also known as glycine, and its pet name can be gly simply. Okay, you can call it gly, right? So please make a note of it. If you have got any doubt, you can ask me. Huh? If anybody is unable to use their audio, they can send the message so that others can ask me on their behalf. Okay, so let us move further. So I was talking about this glycine. So this glycine unit can be combined like this. This is called amino acid. Let me take CH2, NH2, COOH. Okay, so CH2, NH2, COOH. So this is called glycine or gly. Similarly, you can have, this is alpha amino acetic acid. Similarly, alpha amino propionic acid. That is, this is one amino. What about this? This is the COOH group. So from here, okay, so 2 amino. And suppose if it is like this, CH3, CH, NH2, and then COOH. This is called 2 amino and then propionic acid, is it not? So alpha amino propionic acid, that is a common name. IUPAC is 2 amino propionic acid, right? And this can also be called alanine, and its a pet name can be ALA. So this gly and gly can combine to form a gly-gly peptide. Are you able to follow? So the combination of amino acids result in peptide. So this is a dipeptide. It can be a tripeptide, tetrapeptide, pentapeptide, polypeptide. Are you able to understand this? Similarly, gly and ALA and gly can be combined. So this is a trimer. So it can be called gly-ALA gly. So likewise, it can be named. And simply you can put GAG. Okay. Have you noted down? Shall I move to the next one? Gokul. Right. Next. So I was discussing about this. Level pairs get here. So peptide linkage. Okay, so peptide linkage. So this is the one I was talking about. Like a glycosidic linkage, you have peptide linkage. That is the bonding between 2 amino acid units. When they form dipeptide, for instance, glycine, glycine. Is it not so? Gly. So this unit is called what? The peptide, linkage. Similarly, tripeptides, we have got this peptide linkage. So by breaking the higher members of polypeptide, we can get the lower members of what? Peptide, and finally we can get amino acids also. So amino acid is the fundamental unit for this protein. Do you understand? So basically it is an alpha amino acid. So this can be represented like this: NH2, CH2, C double bond O, OH. Okay, right. So we will discuss in detail about this. Then let us introduce the enzyme. As you know, enzymes are biocatalysts. So they are generally chemically proteins, but they are capable of activating a particular reactions. The speciality of biocatalysts is they are very, very bond specific. So that is the very important thing, and that is why we make use of these enzymes very effectively in our day-to-day life in pharmacology, pharmaceutical chemistry, medical treatment, etc. And then vitamins, they are accessory food factors. They are required very much in the diet. Their presence may be the least, but its deficiency leads to many diseases, and that's why it's called vitamin deficiency. Okay. Next one. As I have already discussed the protein, now let us come back to the nucleic acid. That's also very important biomolecule. So after discussing this, let us take up the glucose, fructose, and all these carbohydrates one by one. Now let us consider the nucleic acids. So what are nucleic acids? Nucleic acids are the polymers of nucleotides. No. Nucleotides. Okay, take down. So they are the polymer of nucleotides. It consists of pentose sugar, base, and phosphate. So there are two types of acids here. One is DNA, that is deoxyribonucleic acid. The other one is RNA, ribo or ribonucleic acid. So the different types of monosaccharides. Now let us start with the carbohydrate, and we will explain in detail one by one. Let us take up the sugar, that is carbohydrate first. So what are the different types of carbohydrate? As I already mentioned, they are called saccharides. So let me talk about the monosaccharide first of all. If it has 3 carbon atoms, then you can call it triose. As I already mentioned, ose is sugar, a saccharide. And if it is aldehyde based, then it is called aldotriose. If it is keto, if it has the potential functional group keto, then it's called ketotriose. So similarly for tetrose, aldotetrose, ketotetrose, and pentose, then aldopentose, ketopentose, hexose, so likewise we have. Okay. Then structure of glucose. The structure of glucose can be depicted like this. So truly speaking, it consists of